Hi, my name is Pratik Raikwar, and today I'll be talking about the Kappa opioid receptor system, which I'll be referring to as the KOR, and its clinical potential for alleviating itch. As far as some background here, approximately 15% of the population experiences chronic itch, which is itch that lasts more than 6 weeks. Unfortunately, our knowledge of cures is pretty limited. However, we do know that itch sensory information hits skin receptors, then travels up the dorsal root ganglia, then to the brain through what's called the spinal parabrachial tract here. Little is known, however, about how this pathway is regulated. And so I've been writing a review article about the KOR and how it's a modulatory mechanism for itch sensation for the past few months. You can see detailed here front and center that the main finding of my review is that the KOR provides a promising mechanism to alleviate itch. So one goal of the review is to demonstrate that the KOR is involved in the suppression of itch sensation, and I did this by gathering data from pharmacological studies to demonstrate this proof of concept. The next goal was to provide evidence that this KOR-mediated modulation of itch occurs along the spinal parabrachial tract, which I did by analyzing neuroanatomical studies. Without getting too far into the details of the methods, I basically just searched terms such as KOR and itch in PubMed and screened the papers. And at this time, I have approximately 100 papers in my review, and you can see with this flowchart kind of my screening process. Moving on to the results and discussion section, I thought it was a good idea to combine both of these sections and present it in a timeline here on the right, because I'll be talking about quite a few studies, and hopefully this chronology will kind of give you a better understanding of what the KOR regulation of itch looks like from a kind of historical and chronological point of view. So without much further ado, the first conclusion is that the KOR regulation of itch has a historical basis. In the 1980s, one study found that withdrawal from chronic administration of a KOR agonist called U5488 as a treatment for pain sensation actually resulted in excessive scratching in monkeys. You can see that with this 1987 study. Another study found that a variety of relatively unselective KOR agonists suppressed scratching in rats in a dose-dependent manner, but these studies were largely ignored at the time. And heading into the late 90s, researchers began to postulate that an imbalance of KORs and mu opioid receptors are at play in itch sensation. The reason I bring this up is that mu opioids have been known to induce itching despite their pain alleviating qualities. So if KORs are able to both suppress pain and itch, then KORs may serve as a better therapeutic target than mu opioid drugs. So with these opioid theories surrounding itch, I'll move on to conclusion 2, which is that pharmacological studies implicate the KOR as a mediator of itch with clinical potential. And I do that with pharmacological studies again. So in 2002, a couple researchers named Kamei and Nagase conducted a study where they found that injecting a KOR antagonist called NOR-BNI into mice induced dose-dependent scratching. This was the first study with a selective KOR drug, which suggested KOR mediation of itch sensation. The next year, Alan Cowan and his colleagues built upon these findings and found that an even more selective KOR antagonist called 5-GNTI induced even more vigorous dose-dependent scratching in mice. The reason I bring these two findings up in particular is that it highlights this concept of increased selectivity of the KOR receptor correlating with more profound effects in terms of behavior. So it's important to consider this because KOR agonists in therapeutics oftentimes have crosstalk with the mu opioid receptor system. So by using more selective drugs, you can with more confidence say that the effect you're seeing is due to the KOR system in specific and not like the mu opioid system. So with this same logic in mind, many KOR agonist studies, including nalfurafine, difelikefalin, and nalbuphine, have demonstrated robust and replicable itch suppressing capabilities in many studies and are even in current human clinical trials as anti-itch drugs. And so all of this evidence again supports conclusion 2. The final conclusion is that neuroanatomical studies suggest three loci at which dynorphin acts, which is the neuropeptide of the KOR. In 2011, a researcher by the name of Sadet Inan found that there is CFOS expression at lamina 1 of the dorsal horn in the spinal cord in a KOR antagonist model of itch. This is significant as CFOS expression suggests neural activation, and so if this is happening at lamina 1, and lamina 1 is where the spinal parabrachial tract runs through, then you have reason to believe that KOR antagonism is activating the spinal parabrachial tract. Likewise, when Inan gave mice a KOR agonist in this model, there was less CFOS expression along lamina 1, which suggested that KOR agonists suppress itch through a mechanism along the spinal parabrachial tract. Without getting too far into the details, a clinical trial in Switzerland also found that there is a KOR receptor downregulation and mu opioid receptor upregulation at the skin level in chronic itch patients, which implies that the skin is one potential locus at which KOR is mediating itch. You can see that kind of in this central figure here. In 2014, however, we got our first strong evidence of an endogenous source of dynorphin. Cardone and colleagues discovered B5I interneurons, which project all along the spinal cord, and through behavioral testing found that these neurons use dynorphin to suppress itch. This was significant as it was the discovery of the first endogenous mechanism by which all of the previous KOR-mediated mechanisms I've been talking about are occurring. And so for a few years, many thought that this was the only KOR locus involved, but in 2020, the Basuk lab published a paper about LJA5 neurons, which project dynorphin from the brainstem down to all levels of the spinal cord, just like B5I neurons, in a very robust manner, and through behavioral testing also found that there's a relationship with itch. And so this presented another endogenous loci by which KOR is mediating itch sensation. So you can see B5I neurons here, you can see LJA5 here, and you can see proreceptors at the skin level here. And so with these three main conclusions in mind, we have strong reason to believe that designing drugs that target KOR receptors will have therapeutic effects for these chronic itch and acute itch patients. This has been demonstrated for the past few years, like I said, where nalfurafine, nalbuphine, and difelikefalin have been successful in treating various forms of pruritus. I will say mechanistically, however, we still have a long ways to go. The discovery of both LJA5 and B5I neurons suggests that we may be missing more substrates that are acting along the spinal parabrachial tract and that express KOR. Additionally, we must elicit to what degree LJA5 and B5I neurons are contributing to the anti-itch effects, which can be achieved in conducting additional behavioral studies with lesions of either LJA5 or B5I neurons, and can also do that in many animal models. And when this finally happens, we will have even more promising mechanism by which we can treat itch. With that, I'll end my presentation, and thank you for listening.